Today we're going to talk about how genetic information is transferred. So today we're going to talk about the chemical background behind what we talked about last week in week 3, the heredity and the genetics. In order to do that, we need to talk about DNA, deoxyribonucleic acid. This is a chemical found in the nucleus in which all genetic information for an organism is stored. And it's all about information and the way information is stored. This is the most important concept to understand about DNA. The purpose of DNA is the storage and transfer of information, and we will show you, I will show you how, with the way the chemicals work, that this information is stored and transferred. This molecule was first described by James Watson and Francis Crick, who figured out the actual structure of the DNA molecule and that it formed what's known as a double helix. And they used electromagnetic and electrocrystallographer photographs and knowledge of the energy states of various bonding in order to come up with the double helical structure, which is the most energy efficient way to put these molecules together. So each gene of an organism is part of the DNA molecule that controls a particular trait. So a section of the DNA molecule is called a gene, and that section is the control factor for any particular trait in an organism. Now DNA, when it's in the cell, it has to be stored. And in order to do that, there are proteins known as histones that bind to the DNA in the nucleus. And what they do is help condense the DNA so that it can be stored in the chromatin of the chromosomes. A nucleosome is the term used for a basic unit of DNA packaging. And I'll show you on the next slide the diagram that shows how this works, but it turns out that these are DNA, repeat subunits of DNA in the chromatin, and these are wrapped around the histone proteins. Chromatin is a term used to define a complex of DNA and proteins that forms the nucleus chromosomes, nuclear chromosomes. So these bundles of tightly coiled DNA are the chromosomes that are particularly visible during mitosis and meiosis. So here's what we're talking about. Here's the individual DNA molecule, and this individual DNA molecule is then wrapped around a protein spool, as it were, called a histone. And these protein spools, this wrapping is much like you wrap a thread around a spool of thread. And then each of these protein plus DNA wrapped around it is called a nucleosome. And then these are bound together and then wrapped together in what's called chromatin. And then these chromatin molecules come together and form the chromosome. So how are the nucleic acids, how are they put together? What is their structure? The other molecule that's important is a molecule known as RNA. And DNA stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. And this is a molecule responsible for carrying the DNA message out of the nucleus into the cell in order to produce proteins. And this diagram shows the basic structure for both DNA and RNA. This is DNA because the sugar is deoxyribose. It's a sugar molecule, deoxyribose, a phosphate, and a nitrogen base. And this all this put together is called a nucleotide. And then when you have two nucleotides put together, they're attached by their nitrogen base pairs, which complement each other, and I'll also show you that in a later slide. And so two of the nucleotides together is called a base pair. So the nucleotide is the fundamental component of the nucleic acid, with a sugar molecule, which in DNA is deoxyribose, in RNA it's ribose, a phosphate group, and then a base. And in DNA, the bases, there are 4 bases: adenine, thymine, cytosine, and guanine. In RNA, they're the same bases except uracil is substituted for thymine. So this shows the physical structure of DNA. And the physical structure of DNA is a double helix. And what you have, these outside parts are sugar and phosphate. And then the steps in this ladder, this appears like a ladder, these steps are base pairs in the middle here with the sugar here and here. And so energetically, the best structure that this can take is the double helical structure, which is shown here, which looks very much like a winding staircase. And this is just another type of model to show the same thing. Now, as I said, the double helix is put together and there are base pairs which act as the rungs of the ladder. And the rungs of the ladder are formed by complementary base pairs. And the base pairing is the fact that only one specific base will fit with another specific base. And this is shown here that guanine always pairs with cytosine and adenine always pairs with thymine in DNA. And if we're talking about an RNA molecule, the pair partner for adenine is uracil. And this is what it looks like in a diagrammatic structure. Here's the double helix with these stair steps in the middle. And this is what the stair step looks like. You have the backbone, which is deoxyribose. You have the phosphate, which links these sugar molecules together. And then these rungs are base pairs. And it's always thymine attached to adenine, cytosine attached to guanine, guanine attached to cytosine, and adenine attached to thymine. And this is the way the pairing goes. And it's a structural situation as shown by the fact that here is sort of an angular, while this is a circular structure. And this just shows the same thing in a more different kind of model, and it shows the same pairing that occurs. Now, RNA is different in that it only contains one strand instead of 2, and the sugar molecule is ribose instead of deoxyribose. And RNA contains 4 different types of nitrogen-containing bases. 3 of the 4 are the same as DNA, and uracil is substituted for thymine. And so this is the pairing, and the difference here is that you see the sugar, in this sugar, this oxygen remains, okay? And so this is ribose. If this oxygen is gone, it's called deoxy, meaning missing an oxygen, deoxyribose. Here's the phosphate, and then the base. In this case, the base is uracil. Now, these next 2 slides show tables that show the difference between DNA and RNA, differences and similarities. DNA contains deoxyribose, RNA contains ribose. Both contain phosphate groups. The nitrogen bases are guanine, cytosine, adenine, and thymine in DNA, and then guanine, cytosine, adenine, and uracil in RNA. The DNA molecule forms a double helix, while the RNA molecule is a single-stranded structure with varying shapes. DNA is only found in the nucleus, while RNA can be found in the nucleus, the cytoplasm, and then parts of the structure of the ribosomes. There is only one kind of DNA. There are several kinds of RNA. There are 3 principal types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. DNA is reproduced by a process known as replication, while RNA, which is, we will look at in a minute, the DNA replication is one DNA strand is replicated from another DNA strand. RNA, on the other hand, is produced or transcribed from a DNA strand. Now, there's something that was present in biology for a long time, and it was called the central dogma of biology. And the central dogma of biology stated that all molecular information, biological information, always passed from DNA to RNA, and then from RNA to protein. It was always in that direction and never in any other direction. Replication was the process of producing DNA from DNA, and in that process, DNA is unwound and unzipped from its double helical structure, and then a DNA polymerase comes along and makes a complementary piece of DNA. Now, complementary means that the base pairing is matched so that it's the same DNA. It's a mirror image of the original piece of DNA. This results in the end with two identical strands of DNA, and this process occurs in the interphase of the cell cycle. We were talking about the cell cycle, mitosis and meiosis before. When DNA is replicated, it occurs during the interphase, before mitosis and after, before prophase and after telophase. Now, DNA has a few base pairs at the end of each of its strands called telomeres, and these act to limit the number of times that DNA can be replicated. Each time a DNA strand is replicated, a few telomeres are lost until you get to the point where there aren't any anymore, and that DNA strand cannot be replicated anymore. And this is thought to be part of the aging process, this limitation. And so what happens when the DNA cannot replicate anymore is a process known as apoptosis, or programmed cell death. And this is essentially the idea that the cell becomes old and dies. So here's a diagram to show how DNA is replicated. The first part is you have an original strand of DNA to be copied, and the DNA is unzipped by an enzyme that unwinds the DNA from its helical structure and unzips the base pairs. And so both the unzipping and unwinding occurs initially, and then one strand each goes in a different direction. And then DNA polymerase, in the second step, comes along and begins to copy and produce a complementary strand of DNA. And as you can see, because you get these two strands complement each other, you end up with two strands that are complementary to each other, and so in the end you have two strands that are identical. And then the last part of the process is you end up with two DNA molecules. Each of these DNA molecules are identical to the original DNA molecule. Now, as it turns out, DNA is then used for the production of what's called messenger RNA, and messenger RNA is used to make proteins, so it's the first part of the protein making process. Messenger RNA is made in the nucleus and then is transferred out into the cytoplasm. This process is known as transcription, and then that messenger RNA is used to make proteins in a process known as replication. Make sure for quizzes and tests you know the difference between replication, transcription, and translation. It turns out that with the discovery of viruses, there were a certain set of viruses found that were RNA viruses. RNA viruses are viruses that, when they're done reproducing, only contain RNA. Well, the only way that is possible is if the virus can use the DNA in the host in order to produce new RNA. And the only way you can do that is with an enzyme known as reverse transcriptase. So reverse transcriptase, the discovery of reverse transcriptase proved that the central dogma of biology was incorrect. And you should remember that I may ask a question basically saying what discovery proved that the central dogma of biology was incorrect, and it was the discovery of reverse transcriptase.